Today I'll present the work development of Pegylated Liposomal Amphotericin B for instant treatment of cutaneous leishmaniasis through parenteral and oral administration. This work was developed at the Federal University of Minas Gerais, Brazil, is the results of my postdoc, under the supervision of Professor Frederic Frezat. Well, leishmaniasis is a neglectful tropical disease that has different clinical forms, being an anthropozoonosis caused by the parasite Leishmania and found in tropical, subtropical, and southern Europe. One of the most common forms is cutaneous leishmaniasis, which, despite not being a deadly disease like the visceral forms, causes social and psychological stigmas due to the permanent scars generated by injuries. It is transmitted by sandflies, this one. And in Brazil, in the year of 2022, we had more than 13 cases of the disease. And it is a disease that is found in 98 countries and affects a lot of people. There are few drugs available for the treatment of leishmaniasis, which have several limitations such as toxicity, poor patient adherence to the treatment, and potential of the appearance of resistance. We had liposomal amphotericin B, that is AmBisome, that is the most effective and safer therapeutic agent currently available for treatment of visceral leishmaniasis. However, its efficacy is limited in the cases of cutaneous leishmaniasis and in HIV co-infections. It has limitations about its stability in temperatures above 25 degrees Celsius and a low oral bioavailability. And so in the last decades, much efforts have been devoted to aprimorate these nanoformulations containing amphotericin B. So our hypothesis is that pegylated liposomes may improve the therapeutic efficacy of amphotericin B in comparison to conventional liposomes through prolonged blood circulation time and enhanced stability in gastric fluid. And our objectives was to develop pegylated formulations of amphotericin B, compare its efficacy by parenteral and oral routes in a mirror model of cutaneous leishmaniasis to that of AmBisome. So for this, empty liposomes, conventional and pegylated, was incorporated amphotericin B. And the process of doing these liposomes is protected by a recent field patent. We do these formulations and characterize them for a particle size distribution, amphotericin B encapsulation efficiency, and aggregation state using DLS, HPLC, and circular dichroism. And we can see in this table that both pegylated and conventional liposomes were below 200 nanometers and were monodispersed with a negative surface potential. And we do the HPLC for studying the encapsulation rate, and we saw that both formulations containing amphotericin B was encapsulation rate approaching 100%. In these figures, you can see the cryogenic electron microscopy for the pegylated and the conventional liposomes. We can see uni and oligolamella forms of the vesicles, both below 200 nanometers, being our vesicles slightly larger than the AmBisomes, the commercial form of amphotericin B liposomal. Next, we evaluated the aggregation state of amphotericin B by UV absorption and circular dichroism, and we can see in B that the AmBisome has a double typal signal, indicating that the interaction of the lipids are different from our formulations. This one here. and suggesting that our formulations containing amphotericin B was in a less aggregated state of amphotericin B. We also evaluated the kinetic of the release of amphotericin B and pegylated and conventional liposome. And we can see a much faster release of the amphotericin B in our formulation, suggesting a lower aggregation state of amphotericin B in our formulations. We also evaluate the stability of this formulation in an acidic environment, similar to the stomach ambient. And we can see here that our formulations, our pegylated formulations show a greater stability than the conventional formulation. Next, we evaluated the formulations in Leishmaniasis infected biopsy mice, that is a cutaneous murine Leishmaniasis model. And we, in the first experiment, we treat these animals with 7 doses of the formulations, given a 4 days regime, 5 milligrams of amphotericin B per kilo by parenteral routes, IV or IP. And we can see that the formulation pegylated shows a lesion size that is reduced and parasite suppression in the lesions in comparison to control groups, that is saline and empty liposomes, in comparison to ambisome and conventional liposomes. We can also observe this parasite suppression in spleen too, but only for the pegylated formulation. We also evaluated this formulation given by orally and IP route, 10 doses of these formulations, the same doses, 5 milligrams of amphotericin B per kilo, and 2 days intervals. And we can see also controlling the lesion size growth and parasite suppression. The parasite suppression was evaluated by qPCR in the lesion. And this suppression of parasite load is comparable to those achieved by ambisome given by IP route, and this is for orally administration. And we saw for this experiment that the parasite suppression in spleen was only achieved by IP route formulations. And in this experiment, we also evaluate the toxicity by several markers of renal and hepatic toxicity for the amphotericin B formulations, and only urea showed changes with increased levels in the groups that received IP treatment, that was ambisome and pegylated formulations, suggesting that our formulation orally administration have a low renal toxicity. And as a conclusion, this work reports for the first time that pegylated amphotericin B can improve treatment of experimental cutaneous leishmaniasis by parenteral and oral routes in comparison to ambisome, presumably by through prolonged blood persistence and passive targeting to skin lesions that is really difficult to achieve. And the formulation of amphotericin B in pegylated liposomes is a promising candidate drug for the treatment of cutaneous leishmaniasis and for visceral forms in which the parasite is disseminating by other sites of lesions, several tissues. And I'd like to thank our team and our sponsors. This is Lab Nano. The Professor Fresar is the responsible for the laboratory. We work with liposomal formulations for treatment of leishmaniasis and for fungal infections. We work also with topical formulations and dermocosmetic applications. Recently, we launched to the market, Brazilian market, 2 formulations, dermocosmetics for the treatment of baldness. And we have Professor Pedro Guimarães that works with ionizable lipid nanoparticles for vaccines, for infectious disease, and CAR-T therapy. This is us. Thank you for your attention, and it will be a pleasure to answer your questions.
Thanks so much, Virginia. It's amazing. All 5 speakers stayed on time. This is great. Pressure for the next set. Open for questions. Yeah.
Our differential, sorry, my English, is the encapsulation process of amphotericin B that promotes a formulation with less toxicity, and we can achieve the skin with the conventional formulation that is ambisome. It's not reach the skin. It's directly for visceral forms. It achieves liver and spleen, but it not came from the skin. And we achieved this by our process that is really particular. We use the techniques for control of pH and temperature to work with amphotericin B. That is a critical molecule. It's difficult to work with this molecule. So that's the difference.
Cool. Other questions? I'm going to have, oh, we have another one. Ooh, there.
So I noticed that the zeta potential of the particles were extremely low. It was like minus 30 millivolts almost?
For conventional, the one. Oh.
It was like minus 30, minus 27.
Yeah.
Is that what you were hoping for? Because what I would hypothesize is negative particles would repel negatively charged cells.
We need this charge to the process of encapsulation of amphotericin B. So we put lipids that promote this charge. And without this, I can't talk a lot about it, but we can encapsulate without the negative charge. It's for the molecule.
More secrets. I was going to ask a question about whether this product would be more cost effective, because on your table you said one of the problems with the amphotericin B product is it's just expensive. Is this going to be expensive as well?
Actually, we advanced in this study. We optimized the formulation, changing lipids that are expensive for us, and changing the process of obtaining the vesicles. We do a Bungan classic method in this work, but now we use a cheapest method, and now we starting to scalonate these formulations, and we are open to test the formulations too. We want to aggregate value to the formulations and wanted to do PK and PD studies to, but we changed the lipids, and it's cheaper now.
Okay, perfect. Thanks. Any last questions before we go on to the next session? One last question, Clement.
Can you comment on how you selected the dose in your animal studies? Because I noticed the ambisome didn't work in the animal model, and I'm just wondering if maybe the dose was not appropriate.
These doses. We based on in previous studies we have with this amphotericin B, and what we had in the literature. Usually uses 10 milligrams per kilogram for the 4 days, or in 1 day for 30 days, I think. And as we are encapsulating, we reduce the dose, and it works perfectly for us. And I think it's nice to do this because it will be cheaper to produce using less drug, and it works in this model. And so we test these doses in mouse and in hamsters too. In models of visceral leishmaniasis with these doses with a little differences in the administration doses in the intervals, and works too. I have a poster that it was outside.
Good advertisement. Thanks very much, Virginia.
Okay.